Okay. So welcome everyone, and thank you so much for taking time out to attend our seminar today. I'm Catherine Murphy, for those of you who don't know me, and I serve as the science coordinator for the Middle Rio Grande Endangered Species Collaborative Program. Our speaker today is Dr. Megan Osborne. She's a research professor and evolutionary biologist working in systematics, phylogeography, and population genetics at the UNM Department of Biology and Museum of Southwestern Biology. Megan is an active participant in the collaborative program, and we're very pleased that she could be here today to share her seminar. Her contributions to long-term genetic monitoring of the Rio Grande silvery minnow regularly inform both research and management of wild and captive stocks, so very relevant to what we're doing in the collaborative program. And just a quick plug, be sure to check out her recent paper with Diaz and Turner on the transition from microsatellites to SNP-based microhaplotypes in genetic monitoring, which contains some great news for the monitoring program, and I believe it will appear in Molecular Ecology sometime soon. So with that, I'll turn it over to Megan.
Thank you, Catherine, and thanks everyone for coming to a genetics talk to start your morning off. I appreciate it. All right, so we're going to be talking about genetic monitoring of silvery minnow. Okay, let's see if everything, why don't my slides work? Okay, hold on, here we go. There we go. Okay, so I'm sure most of you are familiar with silvery minnow, but for those of you who are not, we'll just go over a little bit of background. So Rio Grande silvery minnow, or Hibignathus amorus, was historically found in the Rio Grande from northern New Mexico all the way to the Gulf of Mexico. And it was also found in the Pecos River from New Mexico to the confluence of the Rio Grande in Texas. So silvery minnow is a small-bodied and short-lived saprinid fish. It's a pelagic broadcast spawner, and it's the only one of the only member of this guild that still persists in the Rio Grande in New Mexico. Historically, there were several other species of pelagic broadcast spawning minnows in the Middle Rio Grande, including speckled chub, Rio Grande shiner, Tropus orca, but silvery minnow is the only one that is left. Spawning in silvery minnow is typically associated with increases in river flow and temperatures that typically occur in the spring and are associated with snowmelt runoff. So they're particularly susceptible to changes in our climate and reductions in snowmelt. So genetic diversity is an important thing to look at. So populations with higher genetic diversity and larger genetic effective population sizes tend to have much greater evolutionary or adaptive capacity to respond to ecological stresses. And these ecological stresses might include things like a novel disease coming into a population and things like that. So the amount of standing genetic diversity is determined by a range of factors, including both population demography, its history, the effective population size, and certain aspects of life history and natural selection. And so looking at genetic diversity provides us a way to assess the genetic health of a population. And if we find that the population is diminished in diversity, it might suggest that we need to take some more active management in that population. So what the heck is genetic monitoring? Okay, so you might be familiar with genetic studies that just take a single genetic sample and then assess variation. Well, genetic monitoring is like that, except that we're taking multiple samples from the same population over many time points. And this allows us to track genetic diversity over contemporary timescales. Genetic monitoring programs require a consistent methodology across the time series. And for silvery minnow, our time series started in 1999 and goes through through this year, 2022. We missed 2 time steps in 2013 and 2014, and this is because the population collapse did not allow us to collect sufficient samples for analysis. So why would we conduct genetic monitoring silvery minnow? So you guys are all likely familiar that environmental conditions in the Middle Rio Grande are highly variable. And because silvery minnow is short-lived, this results in dramatic fluctuations from one year to the next. And these can span several orders of magnitude. In addition, silvery minnow has been subjected to captive breeding and rearing and augmentation since 2003, so more than 20 years. And these kinds of programs can be beneficial in that they can increase diversity in a population, but there's also the potential for negative consequences. So together, these highly variable environmental conditions, short-lived nature of silvery minnow, and the augmentation program have the ability to affect genetic diversity over contemporary timescales. In addition to this, we compare our genetic monitoring data with demographic data. And this might allow us some insights that aren't really possible with only one of these data sets. So this figure here is data taken from Yakulik et al. 2022. And on the y-axis, we have the estimated abundance of wild-born silvery minnow. And so this is indicated by the triangles and the solid black line. And the dotted line here shows the estimated abundance of, or number of, augmented silvery minnow. And I put this up here really to demonstrate the dynamics that are going on in the population. And so you can see that in some years we have a population that's comprised of roughly equal numbers of wild-born and augmented silvery minnow, while in other years we may have augmented fish dominating the population or wild fish dominating the population. And these are important when we are looking at genetic diversity. In addition, the purple arrow down the bottom here shows the scope of the augmentation program, sort of how long it's been going on. The gray arrow shows our genetic monitoring time series data using microsatellites, and then the red arrows indicate genetic monitoring data using microhaplotype data. Okay, so we're going to be talking mostly about microsatellite data because that's what we have that goes through 2022. So for those of you who are not familiar with microsatellites, these are short repetitive elements found in the DNA. For example, you might have a microsatellite that is an AG, and these are the nucleotide bases. And these nucleotide repeats differ in number from within an individual and between individuals. So these are typically non-coding sequences. They're extremely frequent in the genome. And microsatellite-based studies typically assay variation at tens of loci. They're really highly polymorphic, so that means that they have lots of different alleles, which means they're really good for estimating things like genetic effective population size and population divergence. However, because microsatellites are typically selected based on their levels of polymorphism, so you want to look at highly polymorphic microsatellites, this can lead to ascertainment bias. So that means that you're only sort of characterizing variation at a very variable part of the genome. They're also subject to scoring errors. So if one lab runs a panel of microsatellites, they may score them slightly differently to another one. So there can be some issues there. For silvery minnow, we typically score variation at 9 different microsatellite loci, and these have between 9 and 60 alleles per locus. So there's a lot of power in that. And our data series goes from we have a sample from 1997 and then 1999 through 2022. Okay, so we're currently in the process of transitioning to SNP-based genetic monitoring. So SNPs are substitutions of single nucleotide bases. And I'll be talking about microhaplotypes here. And these are SNPs as well. And it just means you have multiple SNPs at a single place in the genome. And this, looking at microsatellites, gives us a little bit more power than just looking at single biallelic SNPs. So unlike microsatellites, microhaplotypes are found in both coding and non-coding sequences. They're really frequent in the genome. And because of the new technologies that we have, we can assay variation at hundreds to thousands of different loci simultaneously. And so these data sets may be more reflective of genome-wide patterns of genetic variation. So for silvery minnow, we have in our panel that we use to characterize, or in our data that we use to characterize these SNPs, we have just over 5,500 SNPs from 3,000 odd loci. And unlike microsatellites, most of these SNPs are biallelic. So there's just 2 alleles, although there are a few that have more than that. So we have a little less power to measure things like allelic diversity. And the data I'm talking about here spans 1999 through 2018. And we're in the process right now of finalizing a panel that will allow us to assay variation at roughly 300 different loci, and then we'll extend this time series out to 2022, and that's what we'll be using for ongoing monitoring. Okay, so just to make sure everyone is on the same page with some of the terms that we're talking about today. So an individual is considered heterozygous when it has different alleles at one or more loci on the same chromosome. So for example, this individual might be called a big A, little a individual. Heterozygosity is a population level statistic, and this is the proportion of heterozygous individuals in a population. In contrast, we have individuals that are homozygous. So this means that they have the same allele at a particular part of the genome. And for example, this individual might be characterized as big A, big A. Genetic effective population size is the size of a breeding population under idealized conditions. And so that means they meet the assumptions of Hardy-Weinberg equilibrium. So things like equalized sex ratio, random mating, no recombination, stable populations, things like this. And then we're going to be talking about genetic drift today. So this is the random change in allele frequencies from one generation to the next because of sampling error. And by this, I mean that like a small subset of individuals reproduce, and that leads to changes in allele frequency in the next generation. Because in a large population, the signature of drift is there's just not very strong genetic drift. So the allele frequencies in your parental generation are reflected accurately in the progeny generation. But if only a subset of individuals breed, then you can get a shift in allele frequencies from one year to the next. So the effects of genetic drift are that allele frequencies will change and that genetic variation will be lost. The smaller the population, the greater the change in allele frequency due to genetic drift. Okay, so what are we measuring in genetic monitoring programs? So I'm only going to be talking about what we measure for microsatellites and SNPs, although we do assay variation of mitochondrial DNA, but I'm not going to be talking about that today. So we measure gene diversity and heterozygosity. So the effect of a bottleneck on these are that the rate of loss is determined by the genetic effective population size. So when effective size is smaller, variation is lost more rapidly. But because rare alleles contribute very little to heterozygosity, the loss of alleles is not necessarily accompanied by a loss of heterozygosity unless the bottleneck is extremely small. We're also measuring allelic richness. So for microsatellites, this is a very sensitive metric because we have lots of different alleles to track that change in frequency. For microhaplotypes, however, this metric's not very sensitive because most of our loci are dominated by only two alleles. So there's just not much room for change there. So allelic richness depends on both the number of alleles and their frequencies. In a bottleneck, rare alleles are more likely to be lost because of the effects of genetic drift. So you're less likely to sample rare alleles when when a small number of individuals are breeding. So we're also measuring a metric called FST. And so this is a measure of the temporal divergence in allele frequencies. And so this ranges from 0. So your allele frequencies in one temporal sample are the same as the allele frequencies in the next. Or a FST of 1, which would mean that they're completely divergent. So you would have no shared alleles between two different samples. And here we're not talking about, in general, we're not talking about differences between different populations, but we're talking about differences between different temporal samples. And then there's two other metrics that we're going to be talking about a little bit today. So the first of these is IBDF. So this is identical by descent. And so this is a related metric. So the higher the value is indicative of more related individuals in the population. So think of this as an measure of inbreeding. And then we're going to be talking about this G2 statistic. And this is a population parameter that summarizes the variance in inbreeding in the population. And so a higher value of G2 means that you have a population that's a mixture of individuals that have a high inbreeding coefficient and those that are unrelated to each other. And this G2 statistic is important because it's very dependent on demographic history, including population bottlenecks and admixture between slightly divergent populations. The higher values of G2 are indicative of a population with more inbreeding. Okay, so to just talk about genetic effective size a little bit more here. So another way of thinking about effective population size is that it's the number of putative parents and the means and variance in the number of offspring that these parents produce. So when you have relatively few parents contributing lots and lots of offspring, your effective population size will typically be smaller. It's tracked in monitoring programs because effective size can be a really sensitive indicator of population decline, and it can also provide information about the potential loss of adaptive potential and long-term population viability. Effective population size is almost always less than the census population size, but it's typically proportional to the census size of the population. So we're going to be talking about two different measures of variance effective population size or effective population size. So the first of these is variance effective size. So this is measured by allele frequency changes. And if you recall from what I was talking about before, this is really measuring genetic drift. And it's measuring the loss of variation from drift. Because it requires detecting a change from 1 year to the next, it requires two temporal samples to estimate it. When the population is stable, variance effective size and the other metric of effective size, inbreeding effective size, these values will be equal. But when you have a fluctuating population and the effects of population supplementation, these values may not equal and are unlikely to be the same. So the other metric that we're measuring is inbreeding effective size, and this is measured by looking at the non-random association of alleles at different loci. It's measuring the number of parents for the sample generation, so it's offset from variance effective size by year. And we are looking at, it only requires a single temporal sample, and again, when the populations are stable, variance and inbreeding effective size will be equal. But once you start supplementing and when the population changes, these values are unlikely to be the same. So we know our genetic sampling protocol is we go out to the river around this time of the year and we sample unmarked fish from at least 3 localities within each river reach. So Angostura, Isleta, and San Acacia. All of our sampling is non-destructive. We take a small fin clip sample from the fish and then the fish is released and left to go about its business. To date, we've sampled just over 7,000 unmarked fish from the Rio Grande and just over 5,000 captive bred and reared individuals that were released to the Middle Rio Grande. And this sampling of these individuals started basically at the commencement of the augmentation program. We also sample the refugial brood stocks from each of the facilities, but I'm not going to be talking about that data today. So this table here just reflects our sample sizes. So the rows that are in gold indicates the samples that were used for SNP development, and then the sample sizes here show the sample sizes for the SNP development given in parentheses. And as you can see, our sampling is very dependent on basically what the wild population is doing. But in general, we do pretty well. And okay. So moving right along here to results. On the left here, we have the data from the microhaplotypes, and this data set is truncated compared to the full microsatellite data set. And as you can see, neither microsatellites or microhaplotypes really show much change in genetic diversity across the time series. And as I mentioned, bottlenecks, unless they're extremely severe, are unlikely to affect gene diversity. However, our microhaplotypes have improved sensitivity compared to microsatellites, and they do allow us to detect changes in observed heterozygosity across the time series. So circled here with the red circle, it shows a decline in observed heterozygosity from 2009 through 2017, whereas microsatellites don't really detect this change. In contrast, if we look at allelic richness, we don't see a huge amount of change based on our microhaplotypes, and this is because the majority of loci are biallelic. So there's just really not a lot of room to detect that change unless it's a really severe event. However, we can see if we look at allelic richness from 2015 through 2017 and 18, we see a decline detected by microsatellites. And this figure here shows the full time series. So if we look at look at gene diversity, and we don't see a great difference across the time series as far as gene diversity goes, very stable. However, we do see, if we look at observed heterozygosity, we see that augmentation, so where the beginning of the arrow is, stabilized observed heterozygosity and stabilized our allelic diversity once it commenced. And this pattern sort of persisted through about 2016, whereupon we start to see a decline in allelic diversity. And this is here is microsatellite data. And you can see, the dotted lines here and the dashed lines indicate a benchmark value. So we derive these benchmarks every year based on a resampling approach and come up with a value for both gene diversity, observed heterozygosity, and allelic diversity, and say we want to be stay above this value. If we drop below, it suggests that things are not going as well as we would like. Okay, so moving on to microhaplotype-based inbreeding metrics. So on the top here, we have F, which is our relatedness metric. So the higher values indicate more related individuals in the population. So the mean individual inbreeding coefficient increased from 2006 to 2008, and this is a significant increase, and it's remained sort of at this level since then, at least through 2018. And here we have our G2 statistic, and we see 2 increases in this statistic, again from 2006 to 2008, and from 2015 through 2018. And this indicates that we have a higher variance in inbreeding. So this means that within our population, we have individuals that are comparatively inbred, and I don't mean like really, really super inbred, but that inbreeding coefficient is higher. And then we have individuals in the population that are not related to each other. And in the bottom here, we have the value FIS. And so this is measuring the heterozygote deficiency or excess per locus. And so here we see an increase in this value, meaning that we have more homozygous individuals than expected. So moving on to 2 of these statistics that are derived from microsatellites, and this is the entire time series. So we have FIS here on the top, and we see that this value kind of fluctuates across the time series, but we have seen a recent increase in FIS in 2021 and 2022, and this is following a population decline that occurred in 2020. And so this suggests that there's a deficiency of heterozygotes in the population. And on the bottom here, we have the G2 statistic. And so calculating this metric based on microsatellites is not as great because we're looking at relatively few loci. So this metric is less sensitive to change because G2 is calculated based on only 9 loci compared to over 3,000 loci based on SNPs. But I wanted to put this up here just to sort of give you a gist of what's going on. So we have seen a number of increases in this metric across the time series, including in 2021. And increases in this statistic typically are associated that occur in the year following a population bottleneck. So that's when we have a small wild population and more fish are introduced from the hatchery. So this figure up here shows a pairwise FST, and this is only a subset of comparisons, and it's looking only at the comparisons after the augmentation program commenced. And the main point I want you to take away from this is we're interested in looking to see if there's divergence in allele frequencies between our captive stocks and our wild stocks. So in the green box here, we have comparisons between consecutive wild samples, and a value that's 0 means that there's no divergence between those 2 samples. And And you can see for the wild comparison, the majority of these comparisons are not significantly different from zero. However, there are some that do differ a little bit. Our blue box here indicates collections of silvery minnow from wild caught eggs, and these were used extensively in the early part of the augmentation program, where those wild caught eggs are reared up and then released back in subsequent years to augment the population. And here again, we have a number of comparisons that don't differ significantly, meaning those collections reflect the allele frequencies in the wild population. If we look at the orange and the salmon colored boxes, these are comparisons between the wild population and then captive stocks. And so here we see that every single comparison, we see divergence from the wild to the captive lot, meaning that the captive fish are only a subset of the wild population, which is expected. And then consequently, we have allele frequency differences between them. And this will become important as we move along here. So this figure here shows the results of a discriminant analysis of principal components. So think PCA. And here we're looking at the principal components shown on just the first discriminant function, because if you do it in a typical fashion, you just get a big blob of points. So on the left here, we have the microhaplotype data. And as you can see, there's like a shift. We have a collection of purple and like orange, dark orange points on the left. And these are all of the temporal collections that were made before 2015. So 1999 through 2012. After the population bottleneck in 2013 and 14, we see a shift in allele frequencies to the right. And this shift is more notable when you look at the microhaplotype data compared to the microsatellites, but even based on 10 microsatellites, we detect this shift. And if we look at the full data set, this is not just a slightly different way of looking at this, we can see that this shift is maintained. And this pattern here suggests that there's been a shift of allele frequencies associated with the population bottleneck and then restocking the population with captively reared individuals that only have like a subset that don't necessarily accurately reflect that historical distribution of alleles. So a cool thing about genetic data is we can estimate gene flow. And so we took the fish released in a given year and we used that information in combination with the wild sample data to estimate a migration rate or gene flow from the hatchery to the wild population. And so this plot here shows that value, which is shown by the triangles and the dashed line. And then we also are able to show the fraction of the total population from the hatchery based on demographic data. So that's over here on the y-axis. And so that's the ratio of augmented fish to the total number of fish in the population. And hopefully you can note here that the genetic data really pretty accurately reflects that ratio. So we have years where, 2005, 2006, 2007, where these values are almost identical. And there are a few instances where demographic data suggests fewer augmented fish within the population, but the genetic estimates suggest that maybe that value is a little bit higher. And then if you look at the correlation between these two values, so a demographic estimate of the number of augmented fish in the population and a rate of gene flow, we can see that these values are strongly related to each other. So genetic estimates of gene flow are positively associated with the fraction of the population comprised of augmented individuals. Okay, so moving right along to effective population size. So what I just showed you before is important because augmenting the fish or augmenting the wild population with captively bred individuals is basically an additional sort of source of genetic drift. So this is expected to impact our estimate of variance effective population size. And this is because we're measuring the change in allele frequencies between temporal samples. So on the top plot here, we have the variance effective size, which only is looking at the wild samples. So a sample of wild individuals collected in 1 year and then the next year. And then the bottom, we have the effective size of the total population. So this is including that input from the hatchery. And they're indicated by the red points. So the dashed lines here show these sort of estimates of effective population size that are in the conservation genetics literature and are referred to as the 5,500 rule. So these numbers were established in the 1980s, sort of target metrics that a conservation program might wish to try to stay above to prevent adverse genetic effects. And so the 500 number is really looking at the effect of drift and the loss of variation from drift. So in this case, theory basically suggests that you want to stay above a variance effective size of 500 to prevent that loss of genetic variation. And so as you can see here, if you're just looking at the wild samples, I mean, variance effective size has fallen below 500 pretty consistently across the time series. But if you look at if we estimate effective size while accounting for that input of hatchery fish, we are always below that magical number of 500, and we're oftentimes very much below that. And so a small variance effective size suggests that we're likely to have a loss of diversity via genetic drift. And so augmentation with hatchery fish acts as an additional source of genetic drift, and it results in a reduction in the variance effective size for silvery minnow. So as I mentioned earlier, that effective size should be associated, or is often associated, with an abundance of species. And so on the top here, we have a wild abundance. So this is only the untagged fish. And then on the next figure down here is our total abundance. So the wild component of the population and the augmented component of the population. So we see that variance effective size is decoupled from these changes, from abundance estimates. And so this means that increases in abundance of silvery minnow are not accompanied by an increase in the variance effective population size. However, if we look at the ratio, we look at the effective population size and then the fraction of the total population that comes from the hatchery, we see that there's a negative relationship between these metrics. So when the population is largely derived from a more limited pool of captive breeders, we see that the effective population size is reduced, and this is because there's increased genetic drift. And then if we move along to linkage disequilibrium effective size, so this is measuring the number of parents in the current generation, we can see that, hopefully the most notable thing here is that this value is always larger than variance effective population size. And so we've seen 2 declines in linkage disequilibrium effective size in 2004 and in 2015, immediately following that population bottleneck. And again, I'm showing those dashed lines of those limits of any of 50 and 500. And so here the important number is that 50 value. And so this is looking at the increase in breeding in the population. And so ideally you want to stay above an NE of 50. And in the case of silvery minnow, we're always well above that value. And here we are looking at the relationship between linkage disequilibrium effective size and abundance of wild individuals and the abundance of the total population. And we can, unlike variance effective size, we see a positive association between NED and the estimated abundance. And this doesn't matter, this relationship holds regardless of whether or not you're looking at just the wild individuals or the total population size. However, again, we see this same negative relationship between the ratio of augmented to wild individuals in the population, such that when the population is derived from a more limited pool of captive breeders, the linkage disequilibrium effective size is reduced. Okay, so just to sum up the main points here, so genetic diversity for the most part has been maintained despite population declines, but we've recently seen some declines in allelic richness and observed heterozygosity, and we're also seeing some increases in these inbreeding metrics, which may be some cause for a little bit of concern. We've seen a dramatic shift in allele frequency composition of the population when comparing the pre-bottleneck and post-bottleneck populations. We find that genetic effective population size is positively associated abundance when you're looking at inbreeding effective size, which means that this is a good metric for genetic monitoring. We find that genetic estimates of gene flow are positively associated with the fraction of the population that's derived from the hatchery. And we find that when the riverine population is dominated by augmented fish, the effective population size is reduced, which means that we could see an increased loss of diversity. And with that, there's lots of people to thank: the Bureau of Reclamation for funding us, and all of the undergraduates that have helped to collecting the data over the last 20 odd years. Their help is invaluable. And Thomas and the folks at SNARC, Charles Ukulik for his data, all the folks at ASA, the Biopark, and Silvery Minnow Refugium. And with that, I will take questions.
Thank you so much, Megan. You all are welcome to type your questions into the chat, or I encourage you to just ask your question directly, given the complexity of this topic. I don't want to misrepresent your question.
That's awesome. No questions.
Oh, you thought you were going to get away that easily, huh, Megan?
No, it's Andy. Hi, Andy.
Hey, good morning. Thanks. That was a great presentation. I do have a couple of questions. We'll start with one of your slides that had I think it was the FIS and the G2 plots on top of each other. Mm-hmm. Is there a value of the FIS or G2 that once you hit a certain threshold cannot be recovered from?
I don't know. Yeah, so I mean, FIS change. I mean, it really fluctuates from one year to the next, and the same with the G2 statistic. The reason people use that G2 statistic when they're looking at heterozygosity and fitness correlations, and sort of the premise of that is populations or individuals that are more heterozygous are less are more fit, right? So they may have more offspring or something like that. And then when you have individuals that are more homozygous, they're the unfit individuals. And so that's sort of why we look at those values. The G2 statistics really strongly affected by the mixture, we think, between the hatchery population and the wild population. So if you think about when you take a captive stock of individuals and you release their progeny to the wild, right? And then they're going to mix, then in the next year, you have those captive individuals breeding with the wild individuals. And because the allele frequencies differ slightly between those individuals will appear to be more unrelated to each other, right? Mm-hmm. But if you get individuals that were released from the captive stock breeding with each other, then those individuals may be comparatively more related to one another because they were slightly more related to begin with. And that's what gives you a higher value of the G2 statistic. I probably didn't answer your question, but it's complicated. It's pretty complicated. And we're really just sort of getting a handle on kind of the interaction of, the wild and captive on these statistics. What we're really interested to look at is like what these statistics look like based on our panel of SNP markers, and see if this result with microsatellites really kind of holds up when we look at, these more recent collections where we know that there's been, there's kind of a sort of really strong mixture between wild and captive individuals.
Okay, yeah, that helps. I actually have a follow-up question that you kind of instigated in your response. If we're looking at the effective population size of the wild and the wild hatchery plots, I don't know if you want to bring that up. It was, I think, along the 50 and 500 metric that you were talking about. If we're looking at the wild population, that population looks like it's doing fairly well.
Could you bring up that,
but
hang
then-
on, can you bring up that slide, Catherine? Because for some reason my slides went away.
Yeah, let me try to do that while you're.
Okay,
So, yeah, if we're looking at the wild population, it looks like those values are remaining above the 500 level. But when you look at the combination of wild and hatchery, we see a massive reduction in those values below 500 and in some cases pretty close to 50. Is that pointing at a huge hatchery effect? Meaning the hatcheries are all near or below that 50 benchmark?
so, oh, there we go. Hold on, let's get to the right slide. Getting closer. Yep, this one. Okay. Yeah. So basically, the effective size of the total population, I think, is what we really probably should be focusing on, because this is where we have a really strong influence of drift. And you can think about, you know, there's sort of a culmination of factors that are causing this effective size estimated to be pretty small. So you have genetic drift when the population declines to a small number, right? So that's one source of genetic drift. Then the next source of genetic drift is you go in there and you take out a subset of individuals or a subset of eggs from the population and you take them to the hatchery. They're only a subset of the wild population. Okay, so there's that effective drift. And then one, take your individuals that have been reared in the hatchery and then put them back into the river. Only a subset of those individuals is going to contribute. So that's another source of genetic drift. So this effective size estimate is sort of capturing those dynamics. And so the greater the drift, the more likely we are to really have a sort of a change in allele frequencies and the loss of alleles. And if this happens, you know, so early on in the program when we were collecting, hundreds of thousands of eggs, there's less drift, right? Because you have a more representative sample. When you switch to captive breeding of individuals and then releasing their progeny, that's further genetic drift, because you really are only, the captive breeding program, we're breeding like maybe hundreds of individuals. And so that's really going to impact sort of what you see in subsequent generations. Did that help, Andy? Yeah,
that helps. Obviously I see in that bottom plot the pretty linear pattern and flat linear pattern, starting in 2003 all the way to present day. And I'm curious if that's solely, well, obviously not solely, but because we've been heavily augmenting the population, while that plot on the top looks like the wild population is doing okay. And for this statistic, right? So
right. I mean, really, it's the interaction of like repeated, population declines, which affects obviously the diversity contained in the wild population. And then a hatchery population is derived from the wild population. So anything that goes on in the wild is really ultimately going to affect what we have in the hatchery. So if you think about, if we look at the allelic richness plot, for example, which is back maybe a slide or two, you can see that diversity is maintained, and through about, oops, where are we here? Yeah, keep going back. You have to go backwards, I think, Catherine. Back one more. Yep, this one here. Okay, so we can see that on the bottom here, allelic diversity is maintained through 2016. So if you think about the refugial broodstocks we have, the hatcheries hold about 4 years of captive broodstock that are used. And you can see that diversity, so anything released in 2015 and 2016 is going to be derived from those probably 2012 fish. I'm hazarding a guess because there weren't 13 and 14s in the hatchery. And so they maintain that. But then in our more recent collections, the hatchery stocks that we have are really, the diversity that's contained is more restricted to what we had left after these repeated bottlenecks. So as time goes on, as we continually have these bottlenecks in the wild, we're going to lose diversity, right? It's going to be kind of a ratcheting down of allelic diversity that we have in the population. So it's sort of a cumulative effect on the population. The more often we bottleneck the wild population, the less diversity we have represented in the hatchery.
Okay, that helps. And I have one more predictive question for you, but I don't want to take up everybody's time by only my questions. Based on the last couple of years of egg collections data, your genetic data and augmentation numbers, what trajectory do you see our diversity and affected population size trending in the next 2 years? We're talking about a short-lived fish, so we should be able to see some of these effects in the next few years, correct?
Yes.
And what do you see? Do you see us maintaining diversity and an affected population size that will contribute to I don't want to say a stable population, but a population that can rebound from these last bad years?
My prediction is that we'll probably see a decline in allelic diversity and a decline in effective population size. I could be wrong, but my guess is that those things will decline.
Okay, thanks. I have no further questions.
Thank you.
Hey, Megan, how many samples did you collect this year so far?
We have not sampled this year, Joel. I'm not overly optimistic about collections. Thomas has collected me 3 fish, so we're off to a great start.
So to affirm or deny your hypothesis of a potential decline, it may take 2 years to find out, or 2 years of sampling, or one whole more collection effort than this one?
Potentially. I mean, ideal. So if we are able to get even 50 fish this year and genotype them with our SNP panel, maybe we'll be able to detect a decline. But we'll see. We'll have to see.
Thank you.
Okay, Rich, do you want to ask your question?
Yeah, thank you. So thanks for the presentation, Megan. So in the current augmentation plan, the numbers of fish that are released in given areas are driven by the numbers that would be put in the river at that area, that together with the wild fish would equal to 1.0 fish per 100 square meters.
Right.
So that where there are fewer wild fish, there are more hatchery fish that are put on top of those wild fish and vice versa.
Right.
Are we swamping the wild population with that strategy? And should this strategy be rethought?
So I don't think that that's really a problem, but you do make a good point in that in years where the wild population is doing well, like, so we get, relatively high densities sort of across the board, then in those years, I would argue that we don't need to stock, right? So that you're limiting that sort of impact of those hatchery fish. But in other years where, abundances are really low, then yeah, we absolutely need to stock. Otherwise, we're not going to have fish in the river. So, we all understand that captive breeding and augmentation can be problematic. But in this case of silvery minnow and in the environment that it exists, which is highly variable, if we want to have silvery minnow, we are largely tied to augmentation, at least under how the system currently operates. And, over the last 20 years of, mega drought, I mean, in other pelagic spawning species, we've seen these populations blink out because of these huge population fluctuations from one year to the next. Augmentation has sort of protected silvery minnow from this. However, continuing to do this, stops extinction, but there are genetic costs to it. And that's what I think is beginning to show in our data.
Thank you.
Okay, thanks, Rich. Kevin, do you want to ask your question?
Yeah, sure. I mean, I typed it there, Megan. I'm just wondering that, being able to forecast looking forward in wild stock, to carry forward some of the theory and assumptions that underpin your research, and whether or not we can look to a way, and whether there are efforts underway to assess whether or not there's any realized demographic consequences to wild reproducing fish. I'm kind of brand new to the area here in the RGSM world, and so I apologize if this is kind of a redundant, or somebody's already covered this, but this would be helpful, I think, going forward in terms of a forecasting perspective, if you could do that.
So this is not something we've done yet, but this is definitely something that we're beginning to have the tools to be able to do. So we now have or almost have a chromosome level genome assembly for silvery minnow, and having some of this next generation sequencing data, we will be able to start to measure things like mutational load and demographic load in the population. And so I think we're getting to the point where we can actually start to maybe tie changes in the sort of the genetic makeup of silvery minnow to potential sort of demographic consequences. We're not there yet, but I think we're really close to being able to figure out sort of research projects to kind of start to really tackle this. And this is definitely something that we're sort of thinking about.
Thank you.
A little bit of a follow-up on that, Megan, and sorry, Catherine. And that was in 2016, there was a peer review, and I believe Dr. Turner and yourself had a reply, I forget, but would you reply differently to that peer review now, or update it in terms of the new information you've gathered since then?
I guess it depends which particular aspect of the peer review you're talking about.
Based on the new data, not so much the questions, but do you think an update of the replies or response or considerations of that peer review need a revised response in some way? Not that it was your burden, but rather, do you think it's necessary?
I think we want to maybe rethink about how we augment the population, and we also need to rethink about like maybe maximizing the contribution of our captive fish. So either breeding more individuals, collecting more eggs for release. I mean, things like that to really sort of maintain as much diversity as possible in the population. I mean, I think these new markers and stuff are going to allow us maybe a little bit more insight into the population that we didn't really have with, looking at just a panel of a small panel of microsatellites. I think, yeah, just having this extra data, I think is going to help us. And yeah, we should, if we need to reevaluate, let's do it.
Thanks, Joel. Allison, do you want to ask your question? You've typed into the chat.
Yes, so over the last couple of years, not all of the hatchery fish got a VIE tag before they went to the river.
Yep.
So I'm kind of wondering how we're planning to look at, well, I mean, I guess it would have been what, 20 and 21. So how are we going to be able to tell if it was a wild, quote unquote, fish or a hatchery fish?
Okay, so in 2020, at least our genetic sample, we're pretty confident that those were wild individuals because we sampled earlier. So we sampled in October before fish were released. And the fish that we sampled were all basically, they were big fish. And so we're fairly confident those were wild fish. In 2021, we sampled, in our regular sampling window, and nothing was marked. And so our fish, the fish we're calling, they're a riverine collected fish. We can't tell whether or not they're a wild versus a hatchery fish. And in the plots in this talk, you probably maybe didn't notice, and I didn't point it out, but we don't have an estimate of the augmented to the total within the total population for that very reason, that those fish were not tagged.
Yes,
That's why, you know, we've been sort of arguing for having those augmented fish tagged, because that gives us some more power to estimate some of these other metrics. And this year fish will be tagged.
Right, but we're still gonna see a bit of a lasting effect on that, right? Or are we just assuming there was zero survival?
No, yeah, you're right. Yeah, we can't tell. And so I will call anything in my reports will be a riverine collected fish versus a true like wild fish. Yeah, we can't tell.
Right, because even years ago, there was a little bit of an argument as to whether or not any of the fish found in the river could be considered wild already. And
Right.
now it's even further complicated just by those, but yeah, okay.
Yeah, so I mean, I think yeah, we've always considered, you know, a wild fish is a fish that was produced in the river, right? Its parents may have been derived from a hatchery, and that's because, you know, we're stocking the river every year with hatchery fish, and so we can't say this is truly a wild fish, but this is what we've got. So
Thanks, Allison. Anyone else have any pressing questions for Megan? So I have one, and you've touched on this a little bit, Megan, but in terms of management based on genetic monitoring, it seems to me that one of the points of your paper was that these microhaplotype, this microhaplotype method, these SNP methods, are tracking the microsatellite findings from before. So that's a good outcome. But in terms of decision making around management of the species, is this new tool or this new set of tools it's not going to change the way we make decisions, but it's going to allow more flexibility and maybe a faster turnaround on basing decisions on genetic monitoring. Is that safe to say?
yeah, that's safe to say. So yeah, so we can, once we have this panel like fully optimized and we're pretty much there, we will be able to run basically our annual sample of like, you know, 300-400 wild individuals in a single sequencing run. So, and then we're assaying variation at 300 different loci. So yeah, so it'll allow us much quicker turnaround time for getting that data. And it also allows us to, you know, genotype those individuals that might be going out to the river relatively, you know, quickly as well. So yeah, so hopefully it will allow us to make management decisions more rapidly. SNP data also allows us to measure these more accurately, measure some of these inbreeding statistics, which we, you know, using like a small panel of microsatellites, they may not be very accurate, but basing these metrics on, you know, 300 loci may give us sort of additional insight that we didn't really have before.
Well, that sounds like good news to me. So
That's good news. Yeah.
well, thank you. Anyone else have any questions? We have a minute and a half left. All right. Those are great questions, and thank you for fielding them, Megan. All right. Well, if no one else has any other questions, we can end the recording. And thank you all for attending the seminar. And thank you, Megan.
Thank you, everyone, for attending.